Hello everyone. So for my second journal club presentation, it's going to kind of follow along the same path as my first one, which was on liver repair therapies. And for this one, I'll dive into the benefits of using a Chinese herb to improve alcohol-induced heart effects in zebrafish. And the title of this article is Protective Role of Resveratrol Against Alcohol-Induced Heart Defects in Zebrafish Embryos. Okay, so what is this study about? So, the study exposes zebrafish to alcohol at different concentrations, being 1% and 2%. It also, of course, contains a control group, and the point of this experiment is to measure or observe pericardial edema, body length, heart rate, and then a series of 3 mRNA levels for 3 various genes found in myocytes, cells of the heart. And then it uses resveratrol, which is a plant-based component of a Chinese herb called Polygonum cupsidatum, and it's used to determine, the point of the article is to determine if resveratrol has any beneficial effects on pressure overload, heart dysfunction, and myocardial hypertension. And why is this relevant? Well, as we know, fetal alcohol syndrome is prevalent, and the study was trying to determine a safe and effective mechanism for reducing the symptoms of fetal alcohol syndrome, especially those related to the heart and atrioventricular septal defects and arterial deformalities. And then they used zebrafish as a model organism because they have transparent bodies, easy to observe morphological abnormalities. They have short reproductive cycles, so it's quick and effective, and you can get lots of trials in a short period of time. And then it's fairly non-invasive compared to other rodents and model organisms for the introduction of alcohol. The protocol is pretty straightforward, so that's why they chose zebrafish. Okay, what was the hypothesis? So the hypothesis for this article states that resveratrol will have protective effects when observing heart abnormalities in zebrafish exposed to alcohol. And furthermore, it stated that resveratrol will be more effective on zebrafish exposed to lower concentrations of alcohol than higher concentrations of alcohol, meaning that you'll see more improvements and recovery to the heart of the zebrafish if exposed to less alcohol. And then some of the morphological and functional abnormalities that the article took note of were tachycardia, pericardial edema, body length, specifically looking for shortening of the body, and a couple other things that we'll get into. Okay, so this is the first figure for the article, and it's figure 1, and it shows pericardial edema in zebrafish 3 days post-fertilization. And on the left-hand side is images of the fish treated with only alcohol, as well as a control group. Then the figure on the right is the resveratrol treated group at 0.03 grams per liter. That was the concentration for the resveratrol. So, what is pericardial edema? So for this article, it was defined as the excessive accumulation of fluid around the chambers of the heart, and it can be visually seen by a large bubble, transparent bubble around the heart. And how is it observed? It was observed via digital photos from an inverted microscope, And what was concluded? The pericardial edema was found to be more severe in the 2% alcohol-treated groups compared to the 1%, and then of course there was no pericardial edema in the control groups. But nonetheless, both alcohol-treated groups did show signs of pericardial edema. And then that's on the left-hand side. The right-hand side, they added the resveratrol in the 1% group, which is the figure in the middle on the right. There was signs of improvement from the middle figure on the left. For the 1% group, the A plus R is how it's identified there on the graph. They did show signs of partial relievement of the pericardial edema compared to the untreated 1% alcohol-exposed group, but the 2% alcohol-exposed group treated with resveratrol did not have any notable signs of morphological improvements. And from that, the article concluded that the 2% group had too severe of heart defects for resveratrol to be a viable and reliable recovery therapy. Okay, so this is another figure from the article, and figure 2A on the left compares body length, and then figure 2B on the right compares heart rate. So the body length was measured by a calibrated reticule of microscope, and heart rate was measured manually from for 1 minute in each fish. So for the body length, the 1% alcohol had, and the 2% alcohol had comparable body lengths, both shortened. And then the 2% was slightly less than the 1%, but nonetheless both shortened compared to the control groups. And the 1% and 2% had recovered lengthened body lengths when exposed to the resveratrol. So in conclusion, the resveratrol improved body length by increasing body length for both 1% and 2% groups. But on the other side, figure 2B, the heart rate. So the heart rate was lowered in the 1% resveratrol treated group, and there was no significant effect in the 2% resveratrol group compared to the groups just simply exposed to alcohol versus the treated alcohol exposed groups. So the figure 2B heart rate were led to believe that the heart rate was the resveratrol wasn't capable of improving heart rate on the 2% group because of the severity of the heart formation at that point. And here's the last and final figure that was presented in the article. It is figure 3, and it compared relative mRNA levels for 3 heart-related genes at 3 days post-fertilization. So mRNA was extracted using trizol reagent, and data analysis were performed using statistical software called GraphPad Prism 7.0. And so this aspect of the experiment was to investigate the underlying mechanisms of resveratrol on the cardiovascular system by analyzing the expression of 3 various genes, mRNA expressed genes. And So the first gene that was analyzed was BMPR2B, and resveratrol changed the mRNA expression of this particular gene, the first gene in the figure, compared to the alcohol-treated groups, and specifically, it elevated the mRNA expression in the 1% and 2% groups, as you can see. And then similarly in the middle one, HAND2, the resveratrol treatment significantly increased HAND2 mRNA expression in both groups treated by resveratrol compared to the groups just exposed to alcohol. And then lastly, the last, all the way to the right, the TBX5A, this one's a little bit different. The mRNA level increased in the 1% alcohol and resveratrol group, as seen as a similar pattern throughout the rest of the figure, but the way this one varies is there was a slight decrease in the 2% alcohol and resveratrol group compared to that of just the 2% alcohol group. So why is this important? These three genes are critical transcription factors for vertebrate heart development. So the resveratrol, for the most part, upregulated all of these genes, which is good, except that last one, the resveratrol 2%. And that simply just has to do with the fact that that gene did not respond to the resveratrol at a higher alcohol concentration, which could be due to a number of reasons. Just the damage was too far gone to make any changes and adjustments to that. Okay, what were the main conclusions drawn in this article? So, resveratrol can be used as a recovery substance, but only to a certain extent. So, what I mean by that is, as suggested in the hypothesis, the lower concentration of alcohol exposure will be more effective on heart defects and damage reversal compared to the 2%. So the 2% alcohol exposure concentration, the fish exposed to 2% alcohol, didn't have great success with resveratrol because the damage done was too advanced to have any reversible effects. And it was seen that resveratrol prevented atherosclerosis, which is the buildup of fat and cholesterol against the walls of the arteries. Also scavenged free radicals, which free radicals are unstable molecules that can cause damage to cells and DNA, so it kind of put a stop to that. And then it inhibited platelet aggregation, which platelets can be harmful because they can create blood clots, which can lead to strokes and heart attacks. Information obtained through this information was obtained through an analysis of heart-related gene expression, as noted. And to end, the article just provided useful insight into some of the advantages of resveratrol, and how it's able to induce protective effects against heart damage from alcohol exposure. And it just gave us a little bit better insight into a remedy for fetal alcohol syndrome. Okay, future directions for this experiment. So like I previously mentioned, the why behind the TBX5A gene not following the pattern that the other two genes did for the 2% group treated with resveratrol is unknown, and they would need to further analyze this phenomenon to determine a reason for that. And they also do not know the effects of resveratrol on heart defects, so they don't know if there are any underlying issues that resveratrol may cause to the formation of the heart, especially at a young age. These fish were exposed at 3 days post fertilization, so that's very young. And then the effectiveness of resveratrol on alcohol concentrations between 1 and 2% is unknown. So there's, a fine line between 1 and 2% is a large difference in alcohol consumption when talking about that in terms of mothers and fetal alcohol syndrome. So further experiments with 1.5%, different points between 1 and 2% would add more validity to these results. So how can the information I learned from this article, be applied to my research project? Well, there are a number of reasons, different directions I can go with this question, but simple answer is I now have a better understanding of recovery and reversal drugs. I know the one in this article was a plant-based, it wasn't necessarily a drug, a plant-based herb, but it gave me a better understanding of reversal treatment options and therapies. And then it kind of gave me some insight on different things that I could observe, abnormalities to be specific, that I could observe in my own research whenever I introduce my recovery drug, inositol, to determine the effects it has on zebrafish development and behavior to alcohol, which is, this article is alcohol based, and my experiment is also to determine the effects of alcohol on zebrafish. And then there's just, every article each week is just giving me a better understanding of different signs and symptoms associated with fetal alcohol syndrome and what that may look like in a zebrafish, and extrapolating that out to a human, different time points, ages, things of that nature. So I am now interested in maybe doing something similar to this article, observing the pericardial edema, which is something I was already taking note of, but now doing a comparison between the resveratrol, or the inositol in my case, versus the simply alcohol treated groups. And then I also like the heart rate, how they introduced that, because that's something that I could easily implement into my study. And yeah, so that's all. And lastly, this is my reference, and I hope you all enjoyed.